Yes, okay. It's 9 a.m. Pacific time, so I would like to welcome everyone to another Global IMMUNOTALK. I'm here today with the co-organizer of the Global IMMUNOTALKs, Dr. Carla Rodling from Yale University, and our fantastic speaker and dear friend, Dr. Marco Colonna from Washington University. So as always, before we start with the introduction of the speaker and the seminar, I would like to remind everyone which are the goals that we have set for this virtual seminar series. We would like to benefit and inspire immunologists across the world in an egalitarian manner. And we would also like to increase opportunities for scientific learning without traveling. So we would like this seminar series to be accommodating, accessible, environmentally friendly, and very inclusive. And of course, we owe this opportunity to the speakers that have graciously agreed to share their knowledge. And to all of you that not only connect to these talks live or they watch them later in YouTube, but also help this initiative immensely by spreading the word. So each time you tell a friend, you retweet a tweet from our accounts, you send an email with the poster, you are letting somebody else know about this opportunity so they can benefit and be inspired by the excellence in immunology that is shared by the scientists that made the discovery. So thank you so much, and please keep helping by spreading the word. So with that, Carla now will introduce our speaker today, and then we'll proceed with the seminar. Thank you.
Thank you so much, Elina, and ciao, Marco. Today I'm connecting from McAllen, Texas, and this is a small city in South Texas, right next to the border with Mexico. So while I'm here, I truly cannot help but think that in the same way that I'm connecting today from a fairly remote place, we probably have colleagues, maybe even some budding scientists, joining us also from remote places. So I find it extra special today that we will be able to learn directly from the discoverer, directly from Marco, about his science. So with this, let me introduce Marco. I have the real pleasure to introduce Marco Colonna today. Marco is a native from Parma, Italy, where he went to medical school. And after his training, he came to the United States, where he did his postdoctoral training with Jack Strominger. And it was during this part of his career that he discovered inhibitory receptors in natural killer cells. Now you can imagine this was a seminal discovery that really helped us understood how these cells function in self-recognition. Now, I know Marco more through his discovery on how plasmacytoid dendritic cells are large producers of type 1 interference. And Marco, I don't know if you remember, but I cannot forget the first time I met you at a winter immunology conference in Asilomar. Now, this work on plasmacytoid dendritic cells was done during his independent part as a researcher while he was at the Basel Institute of Immunology. And I gather these must have been very interesting times and a remarkable experience for him, where he could think freely without the pressure of raising funds, something that I love to learn more from you, Marco, about. That must have been quite a remarkable experience. Now, it was also during this time that Marco's lab cloned a series of members of the immunoglobulin superfamily receptors, including the TREM receptors. And we will hopefully learn more about them today. And it is through the discovery of TREM receptors that Marco led the way into an entirely new area of research and have shed light on the innate immune response to neurodegeneration, which is the topic of his talk today. Now, currently, Marco is the Robert Roch-Beliveau Professor at the Department of Pathology and Immunology at Washington University in St. Louis, and he has been part of that outstanding department for almost 2 decades. I won't have time to go into details, but for all his seminal contributions, Marco has received numerous awards and honors, including being elected to the National Academy of Science of the United States. So Marco, it's a real pleasure to have you with us today. Thank you so much for being a global immunospeaker today.
Ciao Carla, ciao Elina.
Thank you. Now, before we start, as you know, we really like to get to know a little bit more about our speakers and about their career. Now, with your experience and as you look back in your scientific career, we would love to know what was the scientific discovery that was most influential to you when you started your career? Or what were the scientific discoveries? Maybe there's more than one, but could you tell us a little bit more about it? What were the scientific discoveries that were most influential and why?
Well, when I finished medical school, perhaps one of the most influential work that I came familiar with was the work of Paul Berg. At that time, Paul Berg was at Stanford University, but actually, when I came here at WashU, I realized that he had been here before. And he's done some of his seminal work here at Washington University. So Paul Berg was responsible, among others, for discovering DNA recombination. And his seminal work really was the splicing of foreign DNA into plasmid DNA. So to me, that was very inspirational because it was the first experimental DNA recombination and DNA engineering. And so it was providing the opportunity really to clone genes and to sequence genes and understand, really discover new molecules. And to me, that was very inspirational. In fact, I wanted to become a molecular biology and I started my training in molecular biology.
Right. And you went on to clone so many things.
Yeah. And then, of course, I wanted to apply that to immunology. And so, although it was a little bit late, you know, I arrived when the B cell receptor was discovered, the T cell receptor was discovered by Mark Davis, by Suzumoto Nagawa, but I still wanted to clone genes. And so when I had the opportunity, really, I tried to, I saw this area of innate immunity that was not much explored. At that time, toll-like receptors were not even known. And so I started to clone receptor that I thought could be important for innate immune responses. And that's how I started my career.
Fantastic. And it's been a great career. Thank you so much. We very much look forward to your talk, Marco. Thank you.
Thank you, Marco.
Okay. You have no more question for me?
We'll have many more as you give your talk and later on.
Okay. Okay, good. So I guess let me just try to share my presentation here. Share desktop and let's see here.
Yeah, that's perfect, Marco.
Okay, good.
Excellent, thank you.
Okay, perfect. So first of all, Carla and Nina, thank you very much for inviting me to this forum. It's really a pleasure to be here. It's an honor. And congratulations for this initiative. I think it's a great initiative. And I think we'll have a bright future. So as Carla mentioned, my lab has been interested for a long time in innate immune responses and receptors that control these innate immune responses. Initially, we worked on infections and cancer, but recently we became to the realization that some of the innate immune responses are actually important for neurodegeneration, particularly Alzheimer's disease. And so today, I'd like to review some of our work in this area and also to present some new data on these receptors in neurodegeneration. So these are my disclosures. And I'm sure that many of you know Alzheimer's disease is the most frequent form of dementia in the elderly. It's with the aging of the population, Alzheimer's disease has become a major problem. Just in the United States, 1 person every 8 Americans over the age of 65 develops Alzheimer's disease. And so more than 5 million people in this country are affected by Alzheimer's disease. And Alzheimer's disease causes memory loss and a progressive decline in cognitive functions that ultimately leads to death. And pathologists have known for a long time that in Alzheimer's disease, brains has 2 major lesions. One is the accumulation of amyloid plaques, and one is the intracellular accumulation in neurons of neurofibrillary tangles that cause neuronal loss. And these 2 lesions are paralleled by a glial response, which consists of microgliosis and also astrocytosis. So the culprit of Alzheimer's disease is really abnormalities in the processing, in the proteolytic processing of a protein called amyloid precursor protein, which is expressed by neurons. Now, the amyloid precursor protein is expressed on the cell surface and can be cleaved by 2 different proteolytic pathways. One is the non-amyloidogenic pathway, which is mediated by alpha secretase and gamma secretase. But one is the amyloidogenic pathway, which is mediated by base 1 and gamma secretase. And this pathway leads to the generation of peptides called A beta peptide, 140, 142, which are prone to aggregation. And these aggregates form seed in the brain that propagate in the brain and cause damage. According to the amyloid hypothesis, which is the most common hypothesis, this aggregation of A beta, which forms extracellular aggregates, causes stress in neuron. And stressed neuron then hyperphosphorylate a protein called tau, which is associated with microtubules. And hyperphosphorylated tau then also is prone to aggregation and generates the neurofibrillary tangles that you can see here. And these neurofibrillary tangles then cause neural dysfunction, loss of synapses, neuronal death, and ultimately cognitive impairment. So this model has been supported for many years by early studies on the genetics of this disease, in particular in rare cases of familiar Alzheimer's disease, which are caused by mutation in the APP or mutation in the gamma secretase component, PS1 and PS2, which leads to the generation of A beta peptide. And indeed, these mutations are extremely penetrant. 100% of the people with this mutation develop early on Alzheimer's disease. However, genetic studies in the large population of sporadic Alzheimer's disease have failed to identify this mutation in the general population. In fact, additional DNA polymorphisms have been identified, which are much more frequent but less penetrant. The most common polymorphism is a polymorphism of apolipoprotein E, called ApoE4, which increases considerably the risk of developing Alzheimer's disease. But what caught our attention was that a number of DNA polymorphism associated with Alzheimer's disease are actually affecting molecules which we know are expressed in microglia. TREM2, C33, IMPP5D, MS4A, and also MHC class 1 and MHC class 2. So, as many of you know, microglia are the brain-resident macrophages that were discovered almost 100 years ago by Dario Ortega. They have been found by Florence Genoux, by Miriam Erad, by Frederick Geisman to develop very early on in the embryo by the yolk sac. And so these are very primitive macrophages, and they require 2 major pathways for development. One pathway is mediated by CSF1 receptor, which is important for proliferation and survival of microglia. And a second pathway is a TGF beta receptor pathway, which is important for the differentiation of microglia. Now, microglia is important in the development of the central nervous system and also in diseases. It's important for clearing apoptotic neurons. It's important for clearing myelin debris and facilitating remyelination. Microglia also produce inflammatory cytokines that can activate astrocytes and other brain cells in response to injury. And also, microglia produces neurotrophic factors that are important to sustain neuronal growth and survival. So, as I mentioned, genetic studies have shown that many risk factors for microglia are DNA polymorphism that affect molecules expressed in microglia. And one of these molecules that caught our attention is TREM2. TREM2 is a molecule that my lab cloned back in 1999. It's a member of immunoglobulin superfamily, which is expressed on macrophages. It contains one immunoglobulin superfamily domain, and we and others have shown that this domain has broad specificity for polyanionic ligands. It binds to phospholipids. It binds to apoptotic cells that expose phospholipids. It binds to lipoproteins, HDL, LDL, ApoE. It can also bind to bacterial lipoprotein like lipopolysaccharides. It binds to other polyanions like nucleic acid. And recently, it has been shown that through hydrophobic interaction, TREM2 can also bind to A beta. Now, TREM2 is part of a complex that contains a second molecule, an adapter called DAP12. DAP12 is required for trafficking of TREM2 on the cell surface and for transmitting intracellular signal from TREM2. So DAP12 contains tyrosine-based activation motifs, or ITAM, which upon phosphorylation recruit the protein tyrosine kinase 6. And 6 will then activate a number of downstream signaling pathways that ultimately leads to activation of PI3 kinase, AKT, PLC gamma, intracellular calcium phosphorylation, MAP kinase activation, and also actin remodeling, ultimately activation of cells. So early on, we generated TREM2 deficient mice, and we studied DAP12 deficient mice, and we focused on macrophages in vitro. And my student at the time, Isaiah Turnbull, made a very interesting observation. He noticed that when you culture macrophages from wild-type mice in the presence of limiting concentration of CSF1, macrophages are very resistant and they do not undergo apoptosis. However, if you grow TREM2 or DAP12 deficient bone marrow macrophages in condition of CSF1 deprivation, they immediately undergo apoptosis. You see an increase of percentage of PI positive cells. And so this and other experiments suggested to us that TREM2 has an important function in sustaining the proliferation and the survival of macrophages. Of course, these are cells in vitro. And so the question is, what is the relevance of TREM2 on macrophages in vivo? We know that macrophages are in all tissues, and they have different requirements in different tissues. And so one important contribution to this question came from a study of geneticists led by Elena Peltone. These geneticists were investigating a disease called the Nasu-Hakola disease. The Nasu-Hakola disease is a form of dementia which is quite different from Alzheimer's disease. It's an early onset dementia. People die before 50 years old, and the lesions are completely different. There's no plaques, there's no neurofibrillary tangles, but there's a very strong demyelination of the frontal lobes. And in addition, there is very strong gliosis. In addition, these individuals also develop bone pathology, particularly they have bone cysts. And this geneticist discovered that this rare disease is caused by homozygous mutation of either DAP12 or TREM2. So this study was very important because it told us that even though TREM2 and DAP12 are expressed by all tissue macrophages, in fact, in natura, TREM2 and DAP12 are required by macrophages in the brain, by microglia, and by macrophages in the bone, by osteoclasts. The second connection of TREM2 with dementia came in 2013. At this time, two groups independently discovered that a heterozygous mutation, missense mutation of TREM2, the R47H mutation, is associated with a high risk for Alzheimer's disease. One of these studies were actually conducted by a group which included 3 members of Washington University, particularly Alison Goth and Carlos Cruchaga. And we came to know this work because of them. And so at this point, my student at the time, Yamin Wang, decided to look at the impact of TREM2 deficiency in a mouse model of A-beta accumulation. And the mouse model that we chose was the 5-XFAD transgenic mouse model, which has been developed by Bob Bassars in Chicago. Essentially, this is a transgenic mouse model that expresses human APP and human PS1 carrying different mutations that are responsible for familiar Alzheimer's disease. So when you look at these mice, after 2 months, they develop plaques in the cortex that you can see here stained in red with the antibody 6010. And these plaques tend to expand, to propagate. And after 7 months, you see a significant accumulation of plaques. And you can see that around these plaques in green, you see an accumulation of microglia. And you can also see with certain delay an accumulation of plaques in a different region of the brain, in the hippocampus. So we look at 5-XFAD mice. We look at plaques that you can see here in green, and we look at microglia in red, stained with IBA1, and we could see a clear clustering of microglia around the plaques that are enclosing the plaque. But when we look at the TREM2 deficient mice, we saw a defective accumulation of microglia around the plaques that are then free to spread out. Because of our previous work on the proliferation and the survival of macrophages, we look at whether TREM2 deficiency impacts proliferation of microglia around the plaques. So what you can see here in red is a plaque, and then we stain microglia with IBA1, which is a marker of microglia, and with Ki67, which is a marker of proliferation. And we counted microglia, proliferating microglia, in a certain range around the plaques. And what you can see is that indeed in a 5-XFAD mice, you see a very strong accumulation of proliferating microglia in a very narrow distance around the plaques, about 15 to 30 microns. But in a TREM2 deficient 5-XFAD, you see no accumulation of proliferating microglia. Now, the lack of microglia around the plaques has consequences on the density of the plaques. In fact, in 5-XFAD mice, where you have some microglia around the plaques, you see the plaques are more compact. But in mice that lack TREM2 and where you have no microglia around the plaques, you see the plaque density is decreased. And now the plaques are more filamentous and they tend to spread into the parenchyma. This barrier function was actually highly documented by Jaime Grusfelder at University of Yale, and it was shown to be true not only in this mouse model, but also in human. So because of the ability of the plaques to spread, the plaques are also more neurotoxic. In fact, around the plaques, the axon, the neuronal axons and neuronal dendrites tend to become swollen, tend to become dystrophic. And you can actually measure the neurite dystrophy around the plaques by staining brain section for N-terminus APP. And so what you can see here is a plaque, and you see some swollen axon or neurite dystrophy around the plaques. And what you can see is that TREM2 deficient mice where the plaque is more filamentous, we see more accumulation of neurite dystrophy. So the plaques are more neurotoxic. At this point, we ask the question is why TREM2 is required for a proper microglia response to a beta plaque. And for this, we collaborate with Ido Amit at the Weizmann Institute in Rehovot, Israel. Ido has studied microglia at the single cell level, and he has discovered that microglia It mostly consists of what we call homeostatic microglia, expressed in genes like TMEM118, P2Y12, CX3CR1. But there's a small subset of microglia, which we define as disease-associated microglia that express markers of activation. TREM2 is one of them, ApoE, and other molecules like LPL involved in lipid metabolism, and additional molecules involved in endosome and lysosome, like CST7, CTSD, CTSB, and others. So Ido found that in wild-type mice, most of the microglia is found in this homeostatic cluster. However, in the 5-XFAD mice, where you have A-beta accumulation, you have a shift in the population of microglia, so that now you have more microglia in a different cluster that expresses the DAM markers, ApoE, MHC, SPP1, C11C, CST7, and so on. Now, we provided Ido with single cells, single microglia from 5-XFAD mice lacking TREM2. And what we found was that there's the clear blockade of the activation of microglia, such as you only have a partial activation of microglia, but not the full generation of them. And indeed, we can define two different stages in the activation of microglia, stage 1 and stage 2. Stage 1 is independent of TREM2, but stage 2 is dependent on TREM2. So recently, a very talented student in the lab, Ingrid Zhu, has decided to analyze this phenomenon also by single nucleus RNA sequencing. And the reason why we adopted this technology is because this technology allows you to look at changes not only in microglia, which can be easily isolated from the brain of mice, but also to identify changes in other cells in the brain, neurons, astrocytes, oligodendrocytes, that cannot be easily isolated from brain and analyzed by single-cell RNA-seq. And so in this experiment, we took 5-XFAD mice, TREM2 deficient 5-XFAD mice, and control mice. We isolated the cortex, we isolated nuclei, and then performed single nuclei RNA-seq by 10x genomics. Bioinformatic analysis tells us that these nuclei can be clustered in different population, and microglia clusters in this population setting. So we look at the representation of this cluster across the 4 different phenotypes. And what you can clearly see is that in a 5-XFAD mice, you will have an expansion of cluster 7, which is consistent with the microgliosis. Whereas in TREM2 deficient mice, we have seen no expansion of microglia, again suggesting that TREM2 is required for expansion for response of microglia. We can also subcluster microglia in different small clusters. And one of these clusters, particular cluster 1, is expanded in 5-XFAD mice. And this expansion is blocking TREM2 deficient mice. And when we look at the genes that are expressed in these subclusters, we see that this subcluster expresses DAM gene, CST7, CSF1, LPL. So essentially, this experiment tells us that by single-cell RNA-seq, you can confirm that the generation of DAM during A-beta accumulation is TREM2 dependent. Now, in the interest of time, we have shown that these changes are accompanied by limited changes in gene expression profile of other cells like oligodendrocytes, astrocytes, and neuronal genes. So, of course, one important question at this point becomes, how do these data in mouse relate to human AD? And to answer this question, we obtained brain specimens from individuals with Alzheimer's disease, which either carry common variants of TREM2 or other variants of TREM2, like R47H or R62H, which have been associated with increased risk of Alzheimer's disease. We obtained these specimens from the Rush Alzheimer's Disease Center in Chicago and from our Alzheimer's Disease Research Center at Washington University. We performed single nuclei RNA-seq, and also we also validated some of this data by nanostrings using an expression gene patterns which contains about 760 genes that have been implicated in neurodegeneration. And we analyzed this in a whole brain RNA. So first of all, I'm going to show you the signatures of Alzheimer's disease patient with normal TREM2 allele versus control. Again, we obtain a number of clusters. One of these clusters includes microglia. And when we look at genes that are differentially expressed between Alzheimer's disease and controls, in this cluster, we see genes that are upregulated, genes that are downregulated. And I will just concentrate on the genes that are upregulated. Now, in mouse, we know that DAM microglia, activated microglia, expresses upregulates ApoE, TREM2, AXL, CST7, LPL. And indeed, we find upregulation of the same genes, TREM2, ApoE, MHC class 2, also in human, and we validated this by immunohistochemistry in brain section of Alzheimer's disease samples versus control. Now, we also found additional genes that are upregulated that were not known to be expressed in mouse. For example, chitinase 3-like 1. Chitinase 3-like 1 is a secreted protein that binds chitin and has been previously shown to be highly expressed in the cerebrospinal fluid, and actually has been used as a biomarker for Alzheimer's disease. Now, another important feature of disease-associated microglia in mouse is that we have a downregulation of homeostatic genes, CX3CR1, P2Y12, and TMEM118. Now, we are surprised in human, we found that these genes are not downregulated, and in fact, they're upregulated. P2Y12, CX3CR1, and TMEM118. Now, we noticed that this observation had been previously made in a mouse model of peripheral nerve injury. And it was shown that in peripheral nerve injury, the upregulation of some of this marker is driven by a transcription factor called RF8. Indeed, we find that RF8 is highly expressed in human microglia in Alzheimer's disease, and you can see here some of the RFA in the nucleus. And when we take mouse bone marrow derived cells and we overexpress RFA, we induce the expression of homeostatic genes like P2Y12. On the other hand, if we take mouse bone marrow derived cells from mice that are deficient for RF8, we see a reduction of the level of expression of P2Y12. So essentially, this experiment tells us that the microglia signature in human AD reflects not only a response to a beta plaques, as in mouse, but also reflects a response to neuronal injury. We also look at other signature at other cells in the brain, and we start with astrocytes. So astrocytes are important to promote tissue repair in the brain and also to produce glial scars. And indeed, it was known that in Alzheimer's disease, you have a very strong astrocytosis, which is an attempt to repair the neuronal loss. And so we look at clustering of microglia. We found at astrocytes, we found different subclusters, and we noticed that two subclusters were enriched in Alzheimer's disease and were depleted in control astrocytes. And when we look at the difference in gene expression between these two clusters, we found that these two clusters express matrix molecule, matrix protein, collagen, and NCAN, suggesting that in Alzheimer's disease, astrocytes produce more matrix molecule and produce more repair reaction. And indeed, it was also shown previously that NCAN is also a biomarker for Alzheimer's disease in the cerebrospinal fluid. Another important function of astrocytes is to help the metabolism of neurons. Neurons can utilize lipids for their metabolism upon activation, but they're not particularly good in eliminating free fatty acid. So free fatty acid are accumulate in lipid particles, and then these lipid particles are released and captured by astrocytes. Astrocytes then accumulate these lipid particles and fatty acid in lipid droplets, and lipid droplets are then oxidized by mitochondria, generating reactive oxygen species that have to be detoxified by superoxide dismutases. 3 of these genes that are involved in this process are FAB-P5, which is important in free fatty acid transport, HIL-PDA, which is important in facilitating the storage of fatty acids in lipid droplets, and SOD2 is important for the detoxification of reactive oxygen species. And when we looked at the subcluster of astrocytes, we found that there's a subcluster of astrocytes, ASTO3, which is depleted in AD samples as compared to control samples. And this, actually, this cluster is enriched of SOD2, HIL-PDA, and FAB-5. So essentially, this configuration tells us that because of the neuronal loss, astrocytes are no longer needed for disposal of neuronal metabolic waste. Now, when we look at astrocytes, astrocytes, of course, are important to provide a myelin sheath that can wrap axon and facilitate neuronal transmission. Now, again, we look at different subcluster of astrocytes in Alzheimer's disease and control oligodendrocytes, and we find that 2 subclusters, olig-1 and olig-2, are depleted in Alzheimer's disease versus control oligodendrocytes. And when we look at the genes that are expressed in this cluster, we see that they express myelin oligodendrocyte protein, MOG, and also 1 microRNA, which has been shown to be essential for the differentiation of oligodendrocyte precursor cells into myelin-forming cells. So essentially, again, neuronal loss in Alzheimer's disease reduces the need for axonal wrapping by oligodendrocytes. Finally, we look at neurons, and I'm just going to show you some data we've obtained with nanostring. So here we take the whole brain lysate, and we look at the expression of a number of genes by nanostrings, and we compare AD samples versus control, and we see a strong downregulation of a number of transcripts that are important for neuronal plasticity, neurotropic factor, synaptic vesicle, neurotransmitters, axon guidance, and ionic channels. And obviously, the signature is indicative of neuronal cell death. And I like to point out also that reduced levels of some of these molecules in the cerebrospinal fluid, for example, ADCF1, have been previously found to be markers of Alzheimer's disease. So what we learned from these studies is that glial and neuronal signature in human AD and mouse models are different. Specifically, the human signature are indicative of a response to a beta plaque, but also a response to neuronal injury, whereas the mouse signature in a 5-XFAD model mainly reflect a response to amyloid plaque. And of course, this incongruence is important has to be considered when we test specific pathways in mouse model of Alzheimer's disease. Now, of course, one question remains is about what is the impact of TREM2 variants in microglia and human Alzheimer's disease. And so we analyzed specimens from individuals with Alzheimer's disease that carry the variants R47H or R62H. Again, I'm going to show you a nanostring analysis of these samples. Here, in this volcano plot, we look at the global expression in RNA, lysate of different genes, 760 genes. And we see some genes that are downregulated in individuals that carry the R47H mutation, caspase-1, RF8, HLA-DR, AF1, CIB. All these molecules are expressed in microglia. And so this really tells us that in the presence of a mutation of R47H, you have a reduced microglia activation. So essentially, what we found is that the TREM2 mutation, the TREM2 variants, reduce microglia response to pathology. Now, this reduction is not a very strong reduction, it's a subtle reduction, but we speculate that it has a cumulative effect on microglia through the lifetime of an individual. Now, of course, microglia are not primarily involved in neurodegeneration, but they're important modifiers of disease progression, so they control disease progression. And of course, you need additional risk factors. First of all, age, but also additional genetic factors, perhaps environmental risk factors that are important to drive the development of the disease. But what was important to us was to point out that if we enhance microglia response, we can perhaps delay neuronal damage and disease progression. And this suggests that perhaps pharmacological activation of microglia receptor can provide new therapies for Alzheimer's disease. And so in the last few minutes of my talk, I'd like to tell you about a recent work that is presently in press about potential exploitation of microglia for treating Alzheimer's disease. And we've done this work in collaboration with Alector, who has developed a monoclonal antibody against TREM2, which is now in phase 1 clinical trial. And this is work that was largely done by a postdoc in the lab, Shutan Wong. So we tested this antibody in a human mouse model that we generated some time ago. These are transgenic mice that express either the common variant or the R47H variant of human TREM2. These mice are back transgenic mice that are being crossed onto TREM2 deficient mice to eliminate endogenous TREM2. And then they're being crossed to a 5-XFAD model of Alzheimer's disease. We have shown that compared to the knockout mice, the expression of human TREM2 reconstitute the activation of microglia, as you can see here by looking at the expression of SPP1 and GPNMB. But the expression of R47H actually is not as effective as the common variant of TREM2, suggesting again that the R47H reduces the activity of TREM2. Now, we also look at the impact of these variants of the ability of microglia to cluster around the plaques. In TREM2 deficient mice, we see very little clustering of microglia around plaques in blue here. In mice that express the common variants of human TREM2, we see more microglia clustering around the plaques. And mice expressing the R47H variant show some clustering, which is quantitatively less than in mice expressing the common variant. So we look at these anti-TREM2 antibodies is a variant of these antibodies in a phase 1 clinical trial. And we show that this antibody binds to both the common variant and the R47H variant of TREM2. We check whether this antibody can activate TREM2, whether this antibody is an agonist antibody, using a reporter assay in which we express TREM2. And if we engage TREM2, we induce GFP expression. And as you can see, the antibody is inducing GFP expression in reporter cells that express the common variant of TREM2 or reporter cells that express the R47H variant of TREM2. We also look biochemically whether we can activate phosphorylation DAP12. So here we took bone marrow gland macrophages from mice that express the human common variant or the R47H variant of TREM2. And then we stimulate them with the anti-TREM2 antibody. And then we look by anti-phosphotyrosine at the phosphorylation of DAP12. And you can see that every time we stimulate with a specific antibody anti-TREM2, we see phosphorylation of a band which corresponds to DAP12. Finally, as I mentioned at the beginning of my talk, TREM2 is important for survival of macrophages. And we found that if we culture human monocyte-derived macrophages or mouse macrophages that express human TREM2 in the presence of low concentration of CSF1, we see a very poor survival. And if we now add the antibody, we increase the survival of this cell considerably, both expressed in the common variant or the R47H variant. So in vitro, we can say that the antibody is specific and it can activate TREM2 and can sustain survival of microglia. Now we wanted to see what is the impact of this antibody in vivo. And so we perform a first experiment in which we take mice, either the common variant or the R47H mice, perform one acute injection of a very high dose of the antibody, and then at the end of the experiment, we took the brain, isolate microglia, and perform single-cell RNA-seq. So these are the clusters of microglia that you can see in this experiment. Some of these clusters are homeostatic microglia based on the expression of typical markers like TMEM119. Some of these clusters are DEM microglia, activating microglia based on expression of SPP1, CSF1, CSP7, and so on. One cluster of these cells is interferon responsive. These cells express interferon-induced gene. But what I like to do is to take your attention on two clusters, cluster 9, which includes proliferating microglia in the M phase, and cluster 7, which we call transitioning microglia. When we look at the representation of these two clusters in mice treated with a control IG or mice treated with anti-TREM2 antibody, we find that there's an increase of cells in a cluster 7 and cluster 9, which is induced by the antibody. So essentially, the antibody is inducing an increase of transitioning microglia and increase of proliferating microglia. When we look at the genes that are expressed in these two clusters by gene ontology analysis, we find that cluster 7 expresses a number of metabolically important genes, mostly involved in protein biosynthesis. And when we look at cluster 9, we see that many of genes differentially expressed in this cluster are genes involved in cell cycling, in proliferation. So this experiment tells us that the anti-TREM2 antibody, the agonistic antibody, can drive metabolic activation and proliferation of microglia in vivo. But of course, we finally wanted to know whether injecting the antibodies has any impact on the pathology of 5XFAD mice, either the plaques or the behavior of these mice. And so to do this experiment, we took mice that already have developed plaques at 5 months of age, and then we inject them with weekly injection of antibody for 3 months. And at the end of the 3 months, we perform behavioral tests, and then we look at the pathology in the brain. So as I mentioned before, the presence of microglia around the plaque is important to reduce the toxicity of plaques. Plaques that are very compact, we call them inert plaques, are very little cytotoxic, neurotoxic. These plaques can be detected with methoxy-4. Now, the plaques that we call compact plaques, these plaques also have a compact cores, but they have some halo of filamentous microglia. These plaques are partially neurotoxic. They can be detected with methoxy-4, but they also can be detected with the antibody 6010, which detects filamentous plaques. And finally, we have very filamentous plaque, which can be detected only with the antibody 6010, which binds A beta, but not with methoxy-4, which binds to compact plaques. And so we look at the distribution of these plaques and the impact of the antibody treatment on these plaques. So what you can see here is that those plaques, filamentous plaques, they're only detected with the antibody, but not detected with methoxy-4. And we see that the antibody is reducing the representation of these plaques. Now, we also have inert plaques, which can only be detected with methoxy-4, but they're hardly detectable with 6010 antibody. And we see that antibody treatment is actually increasing the percentage of these neuroplaques that are not neurotoxic. And finally, we look at plaques, which we call compact plaques, which have a core, which is compact, where they have some filamentous a halo around the core. And we see no changes by antibody treatment. So essentially, this experiment suggests that the anti-TREM2 antibody can reduce the amount of neurotoxic plaque and can increase the amount of inherent plaques that have no neurotoxicity. Now, as I mentioned, neurotoxic plaque cause neurite dystrophy, which can be detected with N-terminus APP. And so we look at neurite dystrophy in treated mice and control treated mice. So you can see that in control treated mice, we see a significant accumulation of neurite dystrophy in red here around amyloid plaques in blue. And you can see that in mice treated with anti-TREM2 antibody, we see a reduction of neurite dystrophy around the plaque. And this is actually a quantification. So essentially, the treatment is reducing neurite dystrophy. Now, we can also look at behavior of mice with different tests. This is just one test I want to show you. It's called elevated plus maze. Essentially, mice are given the option to stay in closed arm or open arms, and they're about 1 meter elevated from the floor. So it has been previously shown that 5-XFAD mice, because of their lesion, tend to spend more times in the open arms. You could say that the more risk-taking. You can say that because they're demented, they take chances that a wise mouse would not take. And so what you can see is that when we treat mice with an anti-TREM2 antibody, we reduce the time that they spend into open arms. They reduce the number of times they enter into the open arms. So essentially, they become less exploratory, they become less risk-taking, and they become more wise. So what I show you essentially is that a single injection of an anti-TREM1 antibody can activate microglia, at least in our transgenic mouse model that express the common variant or the R47H variant of TREM2. And this activation induces metabolic activation and proliferation of microglia. And the sustained administration of anti-TREM2 antibody can modify the plaques, reduces filamentous plaques, reduces neurite damage. And there's also some subtle but significant changes in risk-taking behavior and anxiety traits in these mice that parallel reduce pathology. Now, in the interest of time, I haven't shown you that activation of microglia also results in increased surveillance activity, increased ramification of microglia, increased capacity to survey the brain territory. And so the take-home message really of this experiment is that we have shown that if we engage human TREM2 with monoclonal antibody, we can enhance the neuroprotective effect of microglia. We can reduce the neurotoxic effects of the A-beta plaque and the neuron damage. And so perhaps this will be an interesting opportunity for therapeutic treatment of Alzheimer's disease. So I will stop my talk here. I just like to acknowledge my lab. I have a lab of very bright and very hardworking and very engaging individuals. We do different aspects of innate immunity. I just want to highlight the people that work on the project that I presented today. I talk about Ingwe and Shutang, but other individuals involved in this project are Simone, Vincent, Yoon, and Jun Chao. And I also like to acknowledge my long-term collaborators, Marina Chell and Susan Gilfillan. Other individuals in this in my labs are involved in innate responses in innate lymphoid cells and plasmacytoid dendritic cells. I also would like to make some acknowledgement. At Washington University, we collaborate with the neurology department, particularly with David Holtzman and Jason Ulrich. A lot of our single cell and single nuclear RNA-seq is done in collaboration with Max Artyomov and the people in his lab, particularly Amanda Swain and Sarah Anday. I'd like to acknowledge Carla Yede for behavioral studies and Wendy Beatty for electron microscopy. We received a brain specimen essential for this work by 3 different centers: the Rush Alzheimer's Disease Center, the Brain Research Institute in Niigata, and our own research center, and particularly led by Rick Perry. We collaborate with cell signaling technology, particularly Tyler Levy and Sean Bezolet and Richard Shaw. We had a collaboration with the University of Brescia, Luigi Poliani, with NanoStrings, Kelly Miller, and of course we have a base long-term collaboration with Ido Amit and Michal Schwartz. And finally, I'd like to acknowledge the electoral group, particularly Tina Schwab, Noor Mustafa and Arya Tassi, and Arnold Rosenthal for the collaboration on anti-TREM2 antibody. So I'll finish my talk. I will stop here. And I thank you very much for your attention. And thank you, Carla and Elina, for this opportunity. Have a wonderful day.
Thank you, Marco, for such a wonderful talk, and especially for leading the way on fundamental biology that now is the foundation for some novel therapeutic potential approaches. Thank you so much.
Fantastic work, Marco. Thank you so much for sharing. That was, I loved your presentation. So thank you so much. So I will quickly share the screen here just to guide the attendees to ask questions. I'm sure there are many questions on your exciting work, Marco, but the format is that all the questions are via Twitter. And so if you'd like to ask questions to Marco, please search for the account Global IMMUNOTALKS, then find the tweet that says ask questions for Marco Colonna seminar here, and reply to that tweet with your question and mentioning the hashtag Global Immuno. And Marco will be using the account ImmunoSpeaker to answer questions. So thank you also, Marco, for agreeing to do Q&A via Twitter. So with that, I would like to remind you that our next great speaker is the co-organizer of the Global IMMUNOTALK, so Carla Rodley. So hopefully you will be able to join us a week from now to hear from Carla about her research. Okay.
I have to say that when we started organizing this, I thought it was way into the future. Now,
yeah, right, time flies. I know you're next, Carla.
Time has come. I look forward to seeing you all next week. Thanks.
Marco, thanks again for all your effort to present, and see you in Twitter. Okay, bye.
Thank you.
Thank you very much.
Bye, everyone.
Bye-bye.